Hello, everybody, and um, welcome to my first tutorial video I'm doing. It's first going to be on resting membrane potential, which is important to talk about first before moving on to action potentials, just because it is required for action potentials to even happen, to have a sort of resting membrane potential. And what I mean by that is the affinity basically for ions to move across the membrane, essentially, and it's controlled by two major factors. And so let's dive in. It'll make more sense as we go a little bit. So I'll start by drawing a cell, which in this case we're just going to draw a circle. And so first of all, ions are dispersed across the membrane of the cell in different proportions. So, for instance, on the inside of the cell, we'll do potassium. We have a high amount of potassium relative to the outside of the cell. And for sodium, let's do sodium is red. We have a low concentration of sodium inside the cell relative to outside the cell. For calcium, we'll do the blue, same thing, but these are just relative to each other. In all reality, there is a incredibly, that's 2 plus. There's an incredibly higher amount of calcium outside the cell relative to inside the cell. And then we'll do lastly one anion, being the chloride ion, and that has a let's do this orange. This has a also low magnitude inside the cell relative to outside the cell. So this creates a situation that gives us our resting membrane potential, which, spoiler alert, is typically around negative 65 millivolts. Really, it's between like 30 to 90 millivolts, but for the sake of most like textbooks and whatnot, they typically have it at 65. So how this negative 65 millivolts for a resting membrane potential occurs is because of two driving forces of these ions. So we'll take potassium for example, but it applies to all of them in principle. So for instance, the first driving force is due to concentration gradient. And what concentration gradient is basically if you have a high amount on one side and a low amount on the other, the ions are going to want to flow from high magnitude high magnitude to low magnitude. Basically, they want to come to an equilibrium where both sides are dispersed evenly. So in potassium's case, potassium wants to go from inside to the outside of the cell. And then for sodium, outside to inside. And yada yada. But that's only one driving force. The other driving force occurs due to its what we call electrical potential. And so this has to do with the charge of the ion. Potassium being a cation, so it has a positive charge. It is attracted to negatively charged things. And so we do have, for the sake of this example and what is most important in this discussion, chloride anions are within the cell. And as potassium leaves, as you can imagine, you start getting a higher amount higher relative amount of chloride ions inside the cell. And what we really care about is just along this edge. As potassium leaves on this edge, whereas at first you might have had, pluses and occasional minus and just a lot of plus charges. Eventually, as potassium leaves, for instance, you'd get a lot higher ratio of minus charges inside the cell. And potassium's affinity for these minus or negative charges, maybe I should say, drives it to go into the cell. So maybe I'll go from here. Drives it to go inside the cell. So you kind of have this push-pull thing for potassium, where on one side it wants to go outside of the cell because of concentration gradient. It wants to disperse, which is partially due to these cations not wanting to be next to each other, but it's also just something called entropy as well, which we won't get into. But it also wants to go inside the cell due to this electrical potential. And so this creates what we call an equilibrium potential. So each ion has its own equilibrium potential, which is kind of a summation of this push-pull thing. So let's make a little homemade table. So for potassium ions, you get for instance, if potassium was the major contributor to this membrane potential, if it did exactly as it wanted and it wasn't affected by the other ions, you would get a negative 80 millivolt potential. For sodium ions, you would get a 62 millivolt potential. And for chloride ions, let's see, you get, sorry, let me just flip through my notes. You get a negative 65 millivolt potential. And then for calcium, you would get a 123 millivolt potential. Now, like I had already spoiled before, the real resting membrane potential is negative 65 millivolts. So you might think to yourself, well, negative 65, that looks like chlorine quite a bit. So maybe chlorine actually, these chloride ions, excuse me, yeah, let's draw their charges. They maybe are the biggest contributor to this resting membrane potential. Well, in all reality, it's actually potassium that's contributing mostly to this resting membrane potential, which is important for the later discussion in action potentials. But, well, and sodium as well. But essentially, potassium has the highest ability to move across the membrane. We call this permeability. And this is because of, I'm just going to draw them as little boxes. They have potassium leak channels, which allow calcium to flow inside and outside of the cell as it wishes. So hence why it's closer to potassium's potential. Now, sodium does have some slight permeability. Chlorine does. Calcium almost has, for the sake of our purposes, has like no permeability. So sodium and chloride also contribute to why this isn't a perfect minus 80 millivolts. But that is essentially how we get resting membrane potential, is you have all of these having this push-pull of concentration gradients, well, I guess minus calcium a little bit, but push-pull of concentration gradients and electrical potentials, and the average for this for a cell is typically negative 65 millivolts. So that is resting membrane potential.